So thanks for the kind introduction, and also thank you everybody for making it up to this session this morning. I'm going to talk today about, well, actually the title indeed is a different one. It's an exercise in phenotypic screening. Here I put the title Human Tissue Models for 21st Century Therapeutics. They have something in common, of course, right? So I think what we're all about is, you know, the space is called MPS. Organ on a chip is also what we refer to it. But the question is, what is the goal, right? And in our view, the goal is to make a difference in lives of patients, right? So by doing what we do best. And that's building fantastic 3D tissue models to help pharma, you know, develop fantastic drugs. And that's also what we are about as Memetas. And the reason why that's so important is because there are actually so many diseases out there that are unsolved. And then we're talking about typically progressive diseases that are progressive over decades of a patient's life, that are multifactorial in nature, and also very often different between patients. Here is a few examples, right? There's ALS, chronic kidney disease, all kinds of cancers, NASH, IBD. And then we have to ask ourselves the question, okay, why is it that these patients cannot be properly treated or cured? Is there something that we're doing wrong after all these billions and billions of euros that we've been spending in the modern pharmaceutical drug paradigm over the past 30 years or so? Well, we think there is a fundamental flaw in the system. If you develop a drug, there is of course a lot of knowledge from fundamental... Closer? Yeah, okay, good. There's a lot of knowledge from fundamental research, clinical research about those diseases. Yet, if we start developing a drug, the first step is typically reducing the complexity of that disease to a few cells on the bottom of a petri dish or a completely different species, which is an animal model. And that reductionistic step is very understandable because a drug development process costs you 12 years, costs you $2 to $3 billion, right? So the idea of starting simple because it gets really complex down the road is actually a very logical one. Yet by starting off with a reductionistic model that might actually be over-reductionistic, you also give a great bias to what you're doing, right? And you're going to find out if that bias is going anywhere in the right direction only in clinical phase 2 when you start dosing in patients. So it's a huge risk that we take in the very, very first steps of drug development. So that's something we need to change. And I think, and with me I think a lot of people in this room, that organ on a chip and MPS models can really be that change, right? The chips in which we grow our cells in 3D, in an extracellular matrix, supported by a perfusion flow, not just build up of the parenchyma, but also the supporting cells like the vasculature, the stroma, the immune cells, and so on. And make sure that you recapitulate as good as you can the essential disease aspects in that chip, so that in the end, when you're looking for compounds, you're not just curing your model, but you're also curing the real thing, which is the patient. Now, of course, that begs the question, what is a good model? And that's a philosophical one, right? I think we can all talk for hours on that. But we at Memetas think, well, you know, let's just start with a number of basic ingredients that we think should be part of every model. So there's the epithelia, right? The barrier tissue that separates self from non-self, inside from outside. And I think that's a very, very obvious one. You see here a nice example of an intestinal tubule, which is hollow. There's flow through the lumen of the tubule. There's an extracellular matrix here, which you don't see because it's not stained for. We patted it in the center, and there's a second perfusion flow for the basal axis. That's one aspect. Then there's also the stromal component, right? The connective tissue. And it's an interesting part of those tissues, and it has been largely overlooked in the past few decades in the drug development scene. But it's getting a lot of attention these days from pharma. It's really emerging because stroma plays a crucial role in chronic inflammation and in fibrosis. So it is actually not just a filler organ. It's really doing something in there, and I think we're starting to recognize that more and more. And then there's of course the vascular component, right? So vasculature, typically the way to study vasculature is to either in isolated fashion or graft it into mice. And with this type of MPS techniques that you see here, this is an example of the organoplate graft. You can grow that vascular bed, right, with the two blood vessels that sprout, you form this angiogenesis, and then we graft on top of that. In this example, it's an hepatic organoid, adult stem cell derived organoid. And then you can really look at the interaction between that hepatic tissue and the vasculature in a microfluidic setting, which I think is really going to be a game changer in vitro modeling, because vasculature is all over the place, right? Liver really is 50% vascularized, comprised 50% of vasculature, for instance. And then there's the immune system. Immune system is, of course, largely researched by pharmaceutical industry. Everybody does it. But to study it in the correct context, in a 3D system, so in a vascular context, in the context of the target tissue of interest is a different ballgame. And I think that's really where our MPS systems are making a difference. So a good model is really just the start of it, right? The next step that you need to take into account is that you also need to have robust and reproducible assays. And it's something that we, at Metas, it's our bread and butter. We develop those assays for our pharmaceutical clients all the time. This is a great assay that we set up with Sakshi Garg together at Merck, which is actually mimicking the diapedesis process. So that's actually the recruitment of immune cells to the site of inflammation. So you see here in red, it's a blood vessel. Again, it has a lumen. There's flow going through the lumen of that blood vessel. It's positioned against an extracellular matrix gel. And then we administer, in this example, T cells, or it can also be monocytes, to the lumen of that blood vessel. You trigger them with the gradient of, in this example, a chemokine gradient, to undergo that diapedesis process, meaning that first they adhere to the blood vessel wall, then they crawl over the blood vessel wall extravasation, and then they migrate in 3D through the extracellular matrix towards the source of that chemokine gradient, or a tumor tissue or another inflamed tissue of interest. And the nice thing is that because it's all happening in the horizontal plane, right? So the Metas technology is membrane-free. It's layered by surface tension techniques. You've probably seen the talk maybe yesterday of Kristin. You can actually follow each and every step of that immune cell, this onset of inflammation process, and really count all the immune cells individually at each stage, the migration length, the migration velocity, but also ultimately the tumor cell killing if of interest. So this is really great. On top of that, we go a step further, and that's really repeat that thousands of times. And that's because the Metas organoplate platform, as you have seen in, for instance, the talk of Kristin or others this week, is a microtiterplate based platform. So it fits in all the microscopes, and it can be handled by robots. And there is 40 to 96 chips on the bottom of that microtiterplate, which is great because it allows you to do on one single plate your positive controls, your negative controls, your replicas, and your dilution series, which in our view is just the basics of decent science. It becomes high throughput though if you start running it through robots, right? So if you have it in a robot, you can start doing hundreds of plates, and if you do hundreds of plates, you can screen thousands of chips, and you can really start doing phenotypic target discovery and compound discovery campaigns. And I'll tell you a little bit more about it in this talk. So let's just go to an example of how we look at disease and how we think we can help pharma in finding those drugs that are going to make the difference in lives of patients. So just take the example of inflammatory bowel disease, right? We already heard we saw some nice work from Linda Griffith in the former talk, which was really impressive. But if you look at inflammatory bowel disease, there's a lot of hypotheses out there, a lot of potential mechanisms to target with your therapeutic compound. So where to start, right? So there's just a few examples here, right? So there is, of course, the interaction with the microbiome. It might actually be that you want to target the loss of barrier function in your epithelium. You might want to look at the stem cell niche in the bottom of the crypts, the proliferation of these stem cells, the differentiation of these stem cells. You might want to look at the mucus secretion that might be impaired. You might want to look at the role of the stromal component in chronic inflammation. You might want to look at all these immune cells that get recruited and all these cytokines that play a role in the IBD process. So really where to start, right? And we think, what we should be doing is start as comprehensively as possible, either by having everything combined in one model or having at least a lineup of all the individual elements to study in that process to make sure that you are actually start with validating your disease hypothesis before you go to screenable assay. And there's many ways to go about it, right? So I already introduced you the intestinal model. So this is actually a Kako tube, right? So it's a workhorse of pharmaceutical industry, yet it's also a workhorse for us because it's such an extremely robust system that it helps you a lot with optimizing assays. So you can interrogate here the barrier function. You can of course disrupt the barrier function with cocktails of cytokines or toxicants. You can measure it, and I think I want to highlight this one. Because otherwise my colleague Desiree is getting really angry with me that I didn't mention the organoteer. So this is the tear instrument, right? So it's an electrical readout where you access the apical site and the basal site through the wells of the microtiter plate, and you measure the impedance spectrum over that barrier. So you have impedance spectroscopy going over it, which is the tear value, right? Transepithelial electrical resistance value. And you can do that actually then for 128 tubes in one go in under a minute time. But you can also do time lapses because this instrument fits into your incubator and is fully optimized to not disturb the temperature profile or the flow profiles in your incubator. So it's really a first-in-class instrument in tear in general, and I think on top of that, membrane-free, flow supported and so on, right, which is typical for the MPS systems. I stop here the commercial break and I go back to the science. Is this okay? And then of course, so you can add your cytokines. You know, you see here, there's a light here, but you know, you see actually that the tight junctions are really changing if you expose to cytokines, and you can actually see a nice drop in tear value as well. And actually, tear is here a much more sensitive readout than, for instance, a fluorescent tracer marker. So that's Kakos. You can also go to organoids, right? So we work a lot with the adult stem cell organoids that are developed by the group of Professor Hans Klevers at Utrecht in the Netherlands. So basically, you stimulate the LGR5 positive intestinal models. You know, you stimulate the Wnt pathway so that they are going to proliferate, and you can let them differentiate and, you know, develop all the subtypes of the intestinal model, and they typically look like this. So they're like little balls with a lumen, and there's fantastic assays that have been developed. You might have seen the work on cystic fibrosis by Hans Klevers that's really fully personalized and is now being used as a companion diagnostic and give rise to really nice drugs. But you know, you want to have that apical axis. So what we do, we grow it as a tubule, similar to the Kako tubes that you saw before, and you see actually very nicely here also these secondary morphological structures developing. They're very nicely leak tight, they have good tear values, so you can really start using these as well for studying barrier function. The nice thing, of course, of these organoids is that, you know, they have these they have the goblet cells already inherently differentiated, so you have this mucus production. Actually, you can also do that with a Kako model. You just mix in the HC29s or you add the mucus separately, and it really changes the whole game in terms of how sensitive these tubules are, right? So we did actually a great study which was published with Estelas, and Kazu is going to talk after me, where we actually looked into the absorption of drugs over the intestinal barrier, and mucus makes a world of difference here. You can do target discovery and validation. So this is the work we did with Galapagos together, where we compared the genotypes of inflamed versus non-inflamed intestinal organoids. You know, we did target knockdown with adenoviruses to make sure that we can actually reverse an inflammatory effect, or protect basically the barrier from getting disrupted, or reduce cytokine production, which is actually very nicely working. Sorry for that. And then, of course, you want to expand it. I already explained to you the immune cell recruitment assay, which is very, very relevant for IBD as well. And this picture keeps on screwing up, so it's actually completely the wrong picture. This is actually work of Nicole Chigurevsky. You might have seen his talk in the beginning of this week at Roche. The picture really is not relevant for what I'm going to tell you, but I tell you what the picture should have been saying. So they did the intestinal model together with neutrophils, and resident macrophages. So they embedded the resident macrophages in the gel and then looked at neutrophil infiltration and the difference that it had to the disruption of the barrier of the epithelium. So the epithelium was more or less used as a sensor there, which is great work. It's actually published in LabChip. So you know, you can look it up or ask me for the link later on. And then, of course, the next step is really to make it as comprehensive as possible, right? So, and that's actually what we do at Memetas, right? So we source the tissues, we get the blood vessels out, we get the intestinal grips, we get the firulas, we get the PBMCs to really make sure that you have one single patient match cells, and you build these models that look like this, right? Where you have the intestinal tubule, where you have the stromal layer in the gel, and where you have the blood vessel on the other side, and you can then add your immune cells either as resident in the gel or, you know, infiltrating from the bloodstream. Now let's shift a little bit. Maybe good to say we did screens on the intestinal model, but unfortunately not all data at Memetas can be showed publicly at a conference. So let's switch gears and look to another screen that we did, which we are allowed to show publicly, which is related to the angiogenesis process, right? So the angiogenesis, we've discussed a lot, I think, already this week about it. It's a really very nice process that is very nicely captured in the MPS type systems. Also in the Memetas system, so here again you see the gel and the blood vessel that we already discussed earlier, right? So there's the blood vessel, there's the flow through the lumen of the blood vessel. Now you use the basal side to administer a cocktail of angiogenic factors, and the blood vessel starts sprouting and forms these sprouts, and you see here a beautiful confocal image of how these sprouts look like, with a very nicely defined lumen over time. So we actually started looking, okay, can we do this in a high throughput screening setup? And it was actually a very nice intellectual exercise, right? So first of all, you need to make sure that you can automate your data capture. So that means the sprouting length, but also the frequency of the sprouts, you know, but on top of that, we also figured out, okay, you also have inherent toxicology data in there, because you can actually look at the main tube as well. And instead, if your sprouts are gone, but your main tube is also gone, you probably are not looking at a therapeutic effect, but you're probably looking at the toxic effect. So the nice thing is that we could monitor both, so both the efficacy and also toxic effect on the main vessel, so that actually is added value for that assay. So there's data quantification. You, of course, figure out your window, right? So there's the negative controls and your positive controls, and then you want to figure out what your vehicle is. We could inhibit those sprouts with the positive controls, so that's nice. And then we started screening. In this example, it's like 1,500 kinase inhibitor library that we screened, so we see it here. And we screened them in Duplo, so that's about 3,000 chips for the primary screen that were, of course, then taken further in secondary screens. You separate them, right? So there's the untreated vessels, there's the positive controls, negative controls, your window here, and then you have the potential drug candidates, and then the real work starts. So you know, what does this data mean, right? So it's the experiment becomes here actually really that's the easy part. But then understanding what you're doing is really the bulk of the work. I already explained to you that there's both the efficacy data and the toxicity data. So the toxicity data helps already a lot with discarding a lot of these data points as being not so useful. This is actually another picture of showing that we had a very beautiful robust C factor and a very good window. And here you see actually all the pathways that we identified, so they're actually blinded. So I'm not going to talk about specific pathways here. But basically what we found was a lot of hits. So I think the next slide actually, oh, so sorry, this one I had to put in from my director of biology. You know, these are the chips that we've been doing. So really a lot of chips. But we had like 1,500 kinase inhibitors. There's 68 hits coming out of that. 52 that actually had a newly identified anti-angiogenic effect. So a lot of them, quite a few of them could be related back to what was actually known from literature, or some of these compounds were actually being under investigation in clinical trials, or some of them already therapeutically available. But there were also quite a few that had no clinical development, and they are, you know, we're looking into those right now. You know, can we take this further, and you know, how are we combining that with our current business model? That's always an interesting one, I think, for all of us. once you start hitting on these fantastic potential therapeutic candidates. But of course, we are not a pharmaceutical company, right? So we really partner this then, sooner or later. And yeah, that brings me to the end of the game. I'm not sure how I'm doing a time, Jan. But I'm doing time for wrap up. Okay, so this is the wrap up, right? So what we see is pharma has been looking into compounds as their gold treasure for years, right? So the compound was the most valuable aspect that they had. At some point they realized, guys, it's not just about the compound. It's also about the target. The target is really, really important, and that is now completely part of the pharma thinking. What's going on right now, and I think this MPS community underlines it, and also all the pharma people that are here underlines is that, okay, a good target is great, a good compound is great, but you still need to understand how to apply it in disease context. So the disease context is a crucial third piece of the puzzle. And I think that's what we as an MPS community add, and for sure it's definitely the mission of MEMETS to add that third piece of the puzzle. And I think I want to leave it with that. The team. This is a 2019 company outing, but I'm promising you guys the 2022 will be a blast. The team here and that are here, there's Divya, Christine, and Daisy Ray here. So reach out to them if you want to make any orders for the Organoteer instrument. And then there is lots of vacancies in the US in Gaithersburg, our site. You know, so reach out to Kristen. We have a great team over there. You really like it, so reach out. We also have positions in Leiden and Enschede. So yeah, thanks. I'll leave it with that, and thank you for your attention. And